everyone, this is Shraddha Thakkar here. Welcome to my YouTube channel Biology with Shraddha. Today in this video, we are going to talk about DNA replication. Yes, I'll be telling you the 7 important steps involved in DNA replication. So let's get started now. Let us first see an introduction to the DNA replication. DNA, as we all know, is a self-replicating molecule. This means that it can make exact copies of itself. That means it can replicate. DNA replication takes place in the S phase of the cell cycle. So for the ones who have forgotten what the S phase is, let me quickly remind you. We have the G1 phase, then we have the S phase, then we have the G2 phase, and then we have the M phase or the division phase, which again gets back to the G1 phase. So this S phase is actually the synthesis phase where DNA replication takes place. This replication is semi-conservative in nature. This means that one strand of the DNA is old and the other one is new. So we are actually conserving one of the strands. Let us now start with the 7 steps involved in DNA replication. The first step is the activation of the nucleotides. Now we know that DNA is a nucleic acid. It is a polymer of nucleotides. These nucleotides are generally present in the cytoplasm, but they are present in the deoxy monophosphate form. That is, they will be present in the form of deoxy AMP, deoxy GMP, deoxy TMP, and deoxy CMP. So these monophosphate forms have to first get converted into the triphosphate forms. That is, we have to make deoxy ATP, deoxy GTP, deoxy TTP, and deoxy CTP. That's the first step. Next is to find the origin of replication. That is where you start or you initiate the DNA replication. Now this is simply called as the origin of replication. It's a specific point at which the DNA replication starts. In eukaryotes, we have several origins. So there will be many points at which DNA replication can begin. But in case of prokaryotes, there is only a single origin of replication. That means DNA replication will start only at one single point. The third step is incision. Incision simply means creating a cut or a nick in the DNA at the origin of replication. This is done with the help of an enzyme, endonuclease. Very important point. The third thing after we have made the nick or the incision is to unwind the DNA. Now we know that DNA is a double helix. There are 2 strands of the DNA. So the double-stranded DNA has to get converted into the single strands. This is done with the help of the enzyme helicase. Now, unwinding of DNA or converting it into the single-stranded form is also known as melting of DNA. After unwinding is done, this leads to a creation of a tension or a strain in the DNA. Now, the strain can be removed with the help of an enzyme called DNA gyrase, also known as topoisomerase. So what this gyrase or topoisomerase does is that it simply relaxes all the supercoiling that take place after unwinding the DNA. Coiling of the separated strands is prevented with the help of single strand binding proteins. So we have the topoisomerase or the gyrase and the single strand binding proteins coming in picture after the work of helicase is done. Next step is to synthesize the RNA primer. Now we know that DNA replication cannot take place de novo. It requires an RNA primer which will provide it with the free 3' OH end. So this primer is actually synthesized with the help of an enzyme called RNA primase. Then after this, the new strand of DNA can be synthesized by simply continuing or elongating the free 3' OH end provided by this RNA primer. Always note that DNA synthesis takes place in the 5' to 3' direction. So this is how the DNA replication actually takes place. I have drawn this basic diagram for you all to understand. These are the two strands of the DNA which are opening up or which are unwinding with the help of helicase. This is called as the replication fork, where the DNA replication actually starts. So we have these two strands. These are the two parent strands which are getting copied into the new strands. Let us understand how exactly are the new strands synthesized. So there are two strands. One is called as the leading strand and the other one is called as the lagging strand. The leading strand is the one which is made in the continuous way, whereas the lagging strand is the one which is made in the discontinuous way in the form of Okazaki fragments. Both these strands are synthesized with the help of an enzyme called DNA polymerase. So as you can see over here, this is the leading strand which is made in the continuous direction, that is 5' to 3', with the parent strand being in the opposite direction like this way, 5' to 3'. This one is the lagging strand which is made in bits and pieces. It's called as the Okazaki fragments. You always find these fragments on the lagging strand because you have to still make the DNA in the 5' to 3' direction. Next, after you have synthesized the DNA, you have to replace the RNA primer with the DNA. So the RNA primer has to be chopped off and it has to be replaced with short DNA segments with the help of another enzyme called DNA polymerase 1. The Okazaki fragments are then later on joined with the help of the enzyme ligase. So that's how we have made our complementary strands of DNA, and this is only possible because of correct base pairing between the nucleotides. So in the newly synthesized DNA, we have one old strand which worked as the template strand, and the newly formed strand is the complementary strand. So as we discussed earlier, there is one old strand and there's one new strand. That's why DNA is said to be replicated in a semi-conservative manner. So like you can see over here, this one is the parent strand and this is the newly synthesized strand. Similarly, this one is the parent strand and these are the newly synthesized fragments, which are the Okazaki fragments of the lagging strand. I really hope you enjoyed the video and I hope you have understood the 7 steps involved in DNA replication. So this was me, Shraddha Thakkar. Please subscribe to my channel Biology with Shraddha and hit the like button, share my videos, and please tell me if you like them. Thank you.